Okay, so you had homework, structure and function of DNA. And then we're going to talk about DNA replication. It's going to be great. So take a look, compare what you got with what I got. It was just a labeling of a DNA into chromosome diagram. You have to label all of the pieces. Comments, questions, concerns on any of those things. Going once, twice, thrice, sold. Okay, cool. Flip the page. If you got something different than what I got, don't assume that you're wrong. We've seen many times that I too can be mistaken. Okay, you can have other answers for this page. Just, throw your hand up and say, what about this? These were just answers that I gave. So, 2, describe how the following are related to each other. DNA and RNA. At this stage, the only relationship we've talked about is the fact that they are both nucleic acids. If you went to the internet and asked, how are DNA and RNA related? I'm sure there would have been an AI out there that would have given you a long explanation about how RNA is copied from DNA and yada, yada, yada. We haven't talked about any of that. At this stage, the only relationship that you could give me based on what I have covered is the fact that they are both RNA. Sorry, they are both nucleic acids. Both nucleic acids. Adenine and thymine, they're both complementary bases. You could talk about how they're both nitrogenous bases, but the relationship is they are complementary. They are the ones that go together inside a DNA strand. How are complementary base pairings and hydrogen bonds related? Complementary bases connect to each other through the use of hydrogen bonds, which we talked about in the notes. Nucleotides and nucleic acids. Nucleotides make up the structure of a nucleic acid. At this stage, that is the only thing we've talked about. And again, if you've got something different than what I've got, say something. If you're just copying things down, it means you didn't do your homework, it means you got a quiz coming in the next couple of days. It's not going to go well for you. Don't just copy things down. Spend the time to do the stuff. Genes and alleles. We have talked about this as well. Alleles are the specific representation of a gene. So the gene might be, what does your hair do? And the allele is wavy, or the allele is straight, or the allele is wildly curly and untamable. Like there's all sorts of, the allele is the specific representation for a general gene. And in the sequence of nucleotides and proteins, we have not discussed letter F. The only way at this stage that you would have done letter F is by going to the internet and getting an answer that I have not covered. Yes?
I got the nucleotide sequence.
Where did that come from? And, well, that you haven't used the word protein in there anywhere, and you're trying to relate these things to proteins. So, I mean, that's true, but you should also then say, and the amino acids are put together to make a protein. Yes, if you went to Google or ChatGPT or Grok, is it called Grok? Twitter's AI. There's a bunch of them. Gemini, Google's AI. If you went to one of those, they would have given you a totally satisfactory answer. This stage, we haven't talked about it. We will, but we have not talked about it yet. Okay, questions, comments, concerns? Yes, Gemini. D, nucleotides, nucleic acids. So nucleotides, if I go back in my notes, nucleotides were these. This is a nucleotide, and it's strings of nucleotides that give you this structure. They give you half of the structure of a nucleic acid, well, of DNA. RNA will behave a little bit differently, but a nucleotide is the pieces that give you the structure for a nucleic acid. Does that make sense? You may have worded it differently, but that's what you're looking for. Any other comments, questions, concerns? Going once, twice, thrice, sold. Okay, cool. Then, and again, you might have flipped the page. You might have come up with a slightly different answer than I got. Don't assume that just because I didn't write what you wrote that you're wrong. Do not assume this. You got something different, throw your hand in the air and we'll talk about it. So here is a, what is this diagram called? A karyotype. That is the answer to number 1. That is a karyotype. Number 2, why is this image prepared from cells that have stopped during metaphase? Because in metaphase, the chromosomes have formed and are lined up. That's why. Okay, I'm gonna flip the page. Question 3, explain how chromosomes are paired up and how the chromosomes are arranged. Here's how I answered it. I said they're sorted and paired up by size, shape, and centromere location, which is in your notes under the section on homologous chromosomes. Because those are the things that pair. Homologous? Homologous chromosomes are the ones that pair up. Did anyone answer that differently? Okay, in a karyotype, the sex chromosomes are placed last or at the end in all karyotypes. Yes. Sorry, you put what? They would be the 23rd pair. If you said at the 23rd pair, you would be correct, yes. Because technically it would be, they would put them at chromosomes 45 and 46, but the 23rd pair, yes, that's the same thing. Good. What type of information can be gathered from this image about the autosomes? I mean, I looked at this thing. The only thing that I could gather from the autosomes here is that there's nothing wrong with them. Like, there is the correct number. Everything's paired. That's all you can deduce from this image. There's no extra autosomes. There's no missing autosomes. That's it. Autosomes are, and we labeled them in the diagram I gave you on karyotypes, the autosomes are the first 22 pairs. Number 6, what shows that there is variation in the chromosome structure? I just said that the fact that there are different sizes and shapes. I mean, looking at them, you can see that there's different sizes, different shapes. They're bent in different spots. I mean, it's not asking you for an earth sign, or an earth-shattering answer here. Identify whether the person is a genetic male or a genetic female. Okay, this is where there's problems in the karyotype. You're only supposed to have 2 sex chromosomes, and this diagram has how many? 3. But one of the things that we talked about is if a Y chromosome shows up at all on a genetic test, this person would show up male. That Y chromosome takes over. The fact that it's there impacts it. Does this person have any chromosomal abnormalities? Yeah, this person has. What's the problem here? Specifically an extra X chromosome. How many sex chromosomes do they have? 3. How many chromosomes does this person have? 47. When would a mistake show up if the sex chromosomes are the problem? Sorry? No, even before that. Sorry? In when? It's not mitosis. It's in meiosis. In meiosis. So depending on if the problem's in the mom or the... How is that necessary, Malik? Put your middle finger away. Either the problem showed up while the mother who donated the egg was an embryo herself, because that's when meiosis occurs in mom. Before mom's even born, all the eggs are there. Or it showed up in meiosis in dad. That's if there's a problem with the sex chromosome, that's almost certainly when it showed up. It has nothing to do with embryo development. It shows up all the way back in the meiosis that produced those egg and sperm. And then why is the image helpful in making a medical diagnosis? Because if you have bonus chromosomes or missing chromosomes, there is a clear genetic cause for some issues. It's all like is what I wrote here. There could be a whole bunch of issues that a person's experiencing. They do a karyotype, they will see immediately, this is the problem. Okay, that was the homework. Comments, questions, concerns. Okay, if there are none, flip the page. Let's talk about DNA replication. Today we are going to talk about two similar but slightly different processes. Those two processes are DNA replication and, if we have time, protein synthesis. Here we go. So, last year, Science 9, y'all talked about mitosis. During mitosis, you would have talked about interphase and prophase and metaphase, anaphase, telophase. And at the end of interphase would be the cell is just living its life, having its best time. At the end of interphase, your DNA gets replicated. That's all that would have been said in grade 9. It gets copied. They might have said specifically, like, did anyone know what organelle inside the cell does the replicating? Did anyone talk about that in grade 9? Okay, that's cool. It's not part of the curriculum. If you got something bonus, great. If not, that's fine. The ribosomes are responsible for doing some of the copying. But DNA gets replicated. And now that we've talked about the structure of DNA, we can actually talk about how that happens. So, DNA replication is the process of creating, hopefully, an exact copy of a molecule of DNA. That's the goal. You want the DNA to be identical to what came before it. If it's not, you have a problem. Because now the DNA is no longer the DNA of the organism, right? And we said there was something in the neighborhood of, what, 3 billion nitrogenous base pairs? So in a copy of 3 billion things, you want 0 mistakes. So, copying. A cell replicates all of its DNA, its whole genome. I don't know what's funny, gentlemen. We good, Dennis and Josiah? Okay. The whole genome once and only once. It doesn't get copied a bunch of times, it gets copied once. Yes, Malik? Yes, Genome. Genome, I'll zoom in. Genome. It's like GNOME, but with an E in the middle. Yeah, Nome. Genome. A human cell. So, and the other piece to this is, if you recall, it's not just one piece of DNA. How many pieces of DNA do you have in your cells at any given time? I hope not. It should be 46. Because those 46 chunks of DNA condense down into your 46 chromosomes. It's not one long chain, it's 46 smaller chains. So rather than copying one chain once, they're copying all 46 of those things get copied together. So in that way, your body is able to speed up the process. A human cell can copy all of its DNA in a few hours, which is wild to think about, with an error rate. So mistakes get made, it happens, but the error rate is only about 1 mistake per 1 billion nucleotide pairs. 1 mistake in 1 billion pairs. So a chain of DNA, if you took all 46 chromosomes and stacked them all together, there's something like 3 billion pairs. So of the 3 billion, your body is able to, every cell is able to copy your DNA with maybe only 3 mistakes, which is impressive. I don't know if you've ever tried to copy a list of 3 billion things. I'm willing to bet you'd make more than 3 mistakes. I would make more than 3 mistakes. Your body's able to do it really, really well. Each new molecule of DNA serves as a template for the formation of its complementary strand, and I'll explain what that means. Complementary strand. And each new molecule of DNA contains only one strand of the original. and one new daughter strand. Okay, so let me try and demonstrate this. Let's use one of these as an example. So I'm coming over here. Here is, I don't know who this is. Someone was supposed to put their name on this. Nobody did. It was really well done. Yeah, this was whoever folded this did a good job. Here it is. So here's a chunk of DNA. Let's assume that this represents a whole chromosome. So when your body needs to do this is whatever I'll use it. What happens is this DNA gets unzipped. Because when you were folding these things, you saw C went with G, C went with where was it? C went with G, T went with A, C went with G, G went with C, and so you got all these base pairs. What happens is your body unzips this and splits it. And for the sake of this, I'm not going to split it all the way. But your body splits this, it severs this, and say, hey, here's the existing DNA, we're going to split it. We know if this survived, if the DNA, the cell survived, the DNA survived, we know these copies are good. One copy is in the cell, they split it. And they say, okay, cool. We're just going to rip these apart. And now we have two copies that we know are good. Those get set aside. And now, because we know these are good, and we know the code on one side of the DNA, in order to make the copy, a ribosome just has to come along and bring in the correct nitrogenous bases all the way along. So if you know that one strand of DNA looks like this, what's the code going to be on the other side? That is going to get slapped together. If the first one's a G, then we're going to have what over here? A is going to get, a G is going to get, a G, G is going to get C, a C is going to get G, and A is going to get a T. And then this is going to get constructed, and you're going to end up with something that looks like this. You got the first two. Well, the first two is a good start. So psi can be your 1 in 1 billion mistake. Because you got the third one wrong. And then what happens, ignore what I'm about to, ignore what I'm doing behind the curtain. Ignore what I'm doing. So we had the strand of DNA that was there, it got torn apart, and then along comes these ribosomes, they're like, okay, well, we know what one side looked like, let's just sew the other side back together. So which one is this? This goes with this. This one has to be this one. Then these are going to sew together. And now we've got two strands of DNA that now in mitosis are going to get arranged in chromosomes, set along the middle of the cell, pulled apart, and then cytokinesis will rip the two cells apart, and now you have two cells, copies of DNA. Yes?
I don't understand how they split it. So say just split those two.
So these were the originals. Yes, this is your original DNA.
And then you did like another piece from another piece that puts it back together.
Okay, so what happens is, so this gets ripped. And on each one of these, you have workers now. Not actual workers. There are organelles that do this. The organelles are called ribosomes. And so what they do is, so you're a ribosome, and you're a ribosome, and now you each are responsible, because, so I am the nucleus of the cell. I ripped the DNA in half, I gave each of you half. I say, okay, cool. I want the other half. You need to make the other half of that one. You need to make the other half of this one. So that at the end of all of this, I no longer exist, and there are two exact copies of that DNA. So you're like, okay, cool. It's like building Lego. Okay, here's my instructions. I have a G here. Okay, cool. I'm going to go get a C. Click. I have an A. Okay, cool. I'm going to get a T. Click. G means I need a C. Click. And you form the chain one step at a time. Like Lego. And what you end up with is this. One long, but you build it piece by piece, mimicking or aligning with the one that was already built for you. It was like someone built half the Lego set, and you just come along and put together the other half. You being a ribosome. And those pieces, yeah, just like that diagram there. And those pieces, like you ask, okay, so where did the nitrogenous bases come from? Well, your cells have to make those. That's why we eat, to take in those nutrients, because you need the nitrogen, the phosphorus, the sugars to build, among other things, to build the structure of DNA. That's why nutrients are a thing. That's why we're constantly bringing in more nutrients. You just stop eating, your body will continue to do this, but it will take pieces from you. Because there's a hierarchy of importance. Some things we do as human beings are less important than other things. And your body knows this. Instinctively, your body's aware of, okay, that leg muscle, probably not as important as my heart muscle, for example. So if I need proteins, my body is going to start taking from muscles that are less important to continue to build and function the muscles that are more important. Diaphragm's pretty important. The heart's pretty important. If you can't breathe and your heart's not beating, your legs are definitely not important. So that's why when we're stationary for a long time, your muscles start to atrophy. Your body's like, we're not using these. We're going to take that protein, we're going to put it somewhere else. A diagram for that is this. This is what I tried to act out is this diagram. And so this master copy here, this green thing, that I kind of sort of highlighted here. The green is the master DNA that you started with. And then after it splits, the pink part gets copied on like a Lego piece. Clip, clip, clip. So then what you're going to get is you're going to get 2 strands of DNA. One which has half of the original. So let's take this one here. There was an A, a G, an A, a G, a T, a T, a C, and it kept going. It keeps going. I'm not going to give it all. But then on this side, we had a T, and a C, and a T, and a C, and an A. How many did I do? 1, 2, 3, 4, 5, 6, 7, 8. Okay, cool. 1, 2, 3, 4, 5, 6, 7, 8. These two are sides that would go together. Like, look, A with T, G with C, A with T. They got ripped apart, and these pieces came in. What goes with A? Well, T, ribosome would clip that piece in. C, T, C, A, G, G. And then over here, you're going to have another strand of DNA. T, C, A, G, G would go. A, G, T, T, C, C. And now look what we have here. We have 2 identical pieces of DNA. A with T, G with C, A with T, G with C, T with A, T with A, C with G, and C with G. Yes, sir. What happens to your D? Same thing happens to all the rest of you. It decomposes, breaks down, enters the soil. That's why grass grows so nicely in a cemetery.
Okay, so like, you know how like viruses kill stuff?
Yeah.
So like, there's also something to like DNA duplication, like this is like.
A virus uses, from my limited understanding, a virus uses the cell's replication system to replicate more of it. Viruses aren't alive. A virus is just a rogue chunk of DNA. Bacteria are alive, viruses are not. And a virus, the codes, the DNA code there hijacks the existing cell's copying structure and copies more of itself till the cell explodes and all those viruses are flooding through the bloodstream again. And different viruses attack different things. But a virus is not living the way we would say, oh yeah, that bacteria is living. That's why antibiotics work on, an antibiotic is against biotic, life. An antibiotic will kill a bacteria, but does nothing for a virus because viruses aren't alive. Your body needs to deal with a virus on its own, which is why viruses can be so nasty to deal with. That's why immunizations are so important. Because if we can prepare your body to deal with a virus before that DNA chunk shows up by introducing it to DNA pieces that are similar to a virus but not deadly, then when the virus does show up, your body's got defense against it. Hands, comments, questions, concerns. Yes, Malik.
So, immunization is basically virus?
A less dangerous version of whatever you're vaccinating against. So there are vaccinations for bacterium and for viruses. Yeah, so what they do is to develop a vaccine for COVID, for example, they develop a vaccine that codes for the virus without the pieces that allow the virus to replicate itself. And then your body's like, and then they inject you with it. And then your body's like, okay, now I have to deal with this thing, but we've set it up so that now your body's dealing with this thing in a safe and quote unquote safe environment where the majority of the population would be fine. It's like are you a soccer player? Well, what sports do you play? Rugby, football, soccer? I know you play one of them. Which one? Rugby. So you prepare for an actual rugby game aggressively in practice. But in practice, if you practice well, it's aggressive. But in practice, your coach has set it up such that in a way so that it should be a safe environment for you to learn all the things you need to learn to defeat the enemy on Thursday after school or whatever the day is. But can you get hurt during practice? Yeah, sometimes things happen. Just like when your body's preparing for the real deal, you experience symptoms of the illness when you get the vaccine. It's the same idea. You will never get as sick from a vaccine as you would from the OG virus, but your body needs to learn how to fight against it. And so it gives it that kind of practice scenario. Imparis.
Is there any way like the cell can revive without spreading the DNA?
Sorry?
Is there any way that the cell can revive without spreading the DNA?
No. If the cell splits and the DNA is not copied, then there is no instructions in the daughter cell telling it what to do because there's no DNA. That's what the DNA is for. The DNA is the instructions for here's how you survive. If that's missing, the cell, whatever just got copied, doesn't know what to do. It's like you telling me, hey, I copied this book out, and you've written nothing on the inside. I throw away an empty cover because there's no point. Okay, that's as far as we get today. What I want you to do is this. Flip the page. It's nice and easy. This is your homework. This page, what do you mean, wait, what? That page, do that page. All it is DNA replication. In the time that remains and for homework, that's what I want to see. No, P stands for phosphate. D stands for sugar. What is the sugar in DNA? Yes, it is, because the sugar is deoxyribose. This is the backbone. Phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose. I just gave you the answer to letter D. A stands for adenine. C, T, and G are the rest there. That is your homework. When the bell goes, you may also go. In 2 minutes. Get that done. May the force be with you. We will do protein synthesis tomorrow.
Yeah.